Hello, bright learners, and a warm welcome back to Grow English Mastery. I'm Scarlett, and I'm absolutely thrilled to guide you through another fascinating story today. Before we dive into our main topic, let's outline our learning roadmap for this episode. First, we will explore the life and career of Nobel Prize-winning biochemist Jennifer Doudna. Second, we will learn 10 essential vocabulary words that will help you discuss science and innovation. And finally, we will review key grammar, such as the passive voice, which is very common in scientific English. So, with our goals clear, let's begin. We often stand in awe of scientific advancements that promise to change our world. But what about the people behind these incredible breakthroughs? Today, we're spotlighting one such remarkable individual. We are going to explore the groundbreaking work of Jennifer Doudna, who co-developed CRISPR-Cas9, a revolutionary gene editing technology. This tool has been described as a pair of molecular scissors that can edit DNA, the very code of life, with unprecedented precision. Her story is one of deep scientific curiosity, brilliant collaboration, and a discovery that is reshaping medicine and biology. So, please get your notebooks ready. Open your minds to an incredible story of scientific discovery, and let's begin our journey into the world of Jennifer Doudna. Jennifer Ann Doudna was born in Washington, D.C. in 1964. However, she spent much of her youth in Hilo, Hawaii. Growing up surrounded by the unique natural beauty of Hawaii, with its diverse flora and fauna, sparked her curiosity about the living world from a young age. She has mentioned that reading a book called The Double Helix by James Watson about the discovery of the structure of DNA was a pivotal moment for her as a young student, igniting her passion for biochemistry. This early fascination led her to pursue higher education in science. She studied biochemistry at Pomona College in California and then earned her Ph.D. in biological chemistry and molecular pharmacology from Harvard Medical School. Her doctoral research focused on RNA, a molecule closely related to DNA that plays many vital roles in our cells. After her Ph.D., she held several prestigious research positions, including at the University of Colorado Boulder, before joining the faculty at the University of California, Berkeley, where she continues her groundbreaking work as a professor. Professor Doudna's research has consistently focused on the intricate workings of RNA and its interactions with proteins. This deep expertise in RNA biology became absolutely crucial for the CRISPR story. For years, scientists had known about strange repetitive DNA sequences in bacteria, called CRISPR, clustered regularly interspaced short palindromic repeats. They suspected it was some kind of bacterial immune system, a way for bacteria to defend themselves against invading viruses. The key breakthrough came through a collaboration. In 2011, Jennifer Doudna met Emmanuel Charpentier, a French microbiologist, at a scientific conference in Puerto Rico. Professor Charpentier had been studying a specific bacterium and had discovered a molecule called Tray-Crin-A, which played an essential role in the CRISPR system, particularly with an enzyme called Cas9. An enzyme, you might remember, is like a tiny biological machine that carries out specific jobs in cells. Doudna and Charpentier decided to pool their expertise. Doudna's deep knowledge of RNA and Charpentier's insights into the bacterial system. They began working together to understand exactly how the CRISPR-Cas9 system identified and cut viral DNA. Their teams worked intensively, and in 2012, they published a landmark scientific paper that changed the course of biological research. In this paper, they demonstrated that the Cas9 enzyme, when combined with guide RNA, which included the trRNA and a CRISPR RNA, could be programmed to find a specific sequence of DNA and cut it precisely at that spot. Essentially, they had figured out how to harness this natural bacterial defense mechanism and turn it into a powerful, programmable gene editing tool. Imagine having a pair of molecular scissors that you can direct to any specific word in a giant instruction manual, the DNA, and make a precise cut or even change the word. That's the power of CRISPR-Cas9. This discovery was revolutionary because previous gene editing tools were much more cumbersome, expensive, and difficult to use. CRISPR-Cas9 offered a method that was relatively simple, affordable, and highly efficient. It immediately opened up vast possibilities for research and practical applications. Scientists around the world quickly adopted the CRISPR-Cas9 system. In basic research, it allowed them to study the function of genes much more easily by turning them off, turning them on, or altering them. In medicine, the potential is enormous. Researchers are exploring its use to correct the genetic mutations that cause inherited diseases like cystic fibrosis, sickle cell anemia, and Huntington's disease. It's also being investigated for developing new treatments for cancer, for example, by modifying a patient's immune cells to better target and destroy cancer cells. There's also research into using CRISPR to combat viral infections like HIV. Beyond human health, CRISPR technology has potential applications in agriculture, such as developing crops that are more nutritious, resistant to diseases and pests, or better able to withstand environmental stresses like drought. For their transformative work on the CRISPR-Cas9 gene editing system, Jennifer Doudna and Emmanuelle Charpentier were jointly awarded the Nobel Prize in Chemistry in 2020. This was a truly historic achievement, marking the first time that a science Nobel was awarded to two women without a male collaborator. It was a profound recognition of their pioneering work and its immense impact. Now, let's focus on some important grammar structures that are helpful when discussing scientific pioneers and their discoveries. First, the past simple tense. This is crucial for talking about events in a person's life and the timeline of discoveries. Jennifer Doudna was born in 1964. She studied biochemistry at Pomona College. Doudna and Charpentier published their groundbreaking paper in 2012. Her early experiences in Hawaii sparked her interest in biology. They received the Nobel Prize for their work. Try to describe key events in someone's biography using the past simple. Second, the present perfect tense. We use this to talk about past actions or experiences that have a strong connection to the present or achievements that continue to have an impact. CRISPR-Cas9 has transformed biological research. Jennifer Doudna has made significant contributions to biochemistry. Scientists have used this technology to study many different organisms. Third, the passive voice. This is very frequently used in scientific contexts, where the focus is often on the discovery or the process itself, rather than who performed the action. The CRISPR system was discovered in bacteria. DNA can be edited with high precision using this tool. The Nobel Prize in Chemistry was awarded to Doudna and Charpentier. Many genetic diseases are being researched using CRISPR. Fourth, modal verbs for expressing possibility, potential, and ability: can, could, may, might, will. These are essential when discussing the future implications of scientific discoveries. CRISPR technology can be used to cure genetic diseases. It could revolutionize agriculture. Further research may uncover new applications for gene editing. This discovery will likely lead to many new therapies. Let's explore some effective techniques to practice your English in the context of science and biography. For listening, seek out interviews with Jennifer Doudna or documentaries about CRISPR. Many universities and scientific organizations also produce podcasts or videos explaining complex topics in accessible ways. Listen for the main ideas, key terms, and how information is structured. For speaking, try to explain to someone in your own simple words who Jennifer Doudna is and why her work is important. What is CRISPR-Cas9? How does it work, in basic terms? Discussing these ideas helps reinforce your understanding and improves your fluency. For reading, look for articles about Jennifer Doudna, CRISPR, or general articles about women in science. Websites of science magazines or university news sections often have profiles or simplified explanations of research. Note new vocabulary and observe how scientific achievements are described. For writing, write a short biography of Jennifer Doudna based on what you've learned, or write a few sentences about what you think is the most exciting potential application of CRISPR technology. This helps you practice summarizing information and using new vocabulary and grammar. Now, let's explore 10 important vocabulary words related to today's topic in more detail. Biochemist noun. A scientist who studies the chemical processes and substances that occur within living organisms. Spelling: Biochemist. Jennifer Doudna is an American biochemist renowned for her work on RNA and CRISPR. A biochemist might study how cells produce energy or how genetic information is passed on. Many biochemists work in research labs in universities or pharmaceutical companies. Gene editing noun phrase: the process of making specific changes to the DNA of a cell or organism. Spelling: Geneditting. CRISPR, Cas9 is a revolutionary tool for gene editing. The potential applications of gene editing in medicine are vast. Ethical considerations are very important in the field of gene editing. DNA noun: deoxyribonucleic acid, the molecule that carries the genetic instructions for the development, functioning, growth, and reproduction of all known organisms and many viruses. Spelling: DNA. Jennifer Doudna's work allows scientists to make precise changes to DNA. the structure of DNA, the double helix, was discovered by Watson and Crick. Each person's DNA contains a unique genetic code. RNA noun, ribonucleic acid, a nucleic acid present in all living cells. Its principal role is to act as a messenger carrying instructions from DNA for controlling the synthesis of proteins, although in some viruses RNA rather than DNA carries the genetic information. Spelling: RNA. Jennifer Doudna has extensive expertise in the study of RNA. Guide RNA is a key component of the CRISPR-Cas9 system, directing the enzyme to the target DNA. There are different types of RNA in cells, each with specific functions. Enzyme noun, a protein that acts as a biological catalyst, speeding up chemical reactions in living organisms. Spelling: ENZYME. The Cas9 enzyme acts like molecular scissors to cut DNA in the CRISPR system. Digestive enzymes in our stomach help break down food. Many industrial processes use enzymes to improve efficiency. Molecule noun, a group of two or more atoms held together by chemical bonds. Spelling: MOLECULE. DNA and RNA are complex molecules essential for life. Water is a simple molecule made of 2 hydrogen atoms and 1 oxygen atom. Biochemists study the interactions between different molecules in cells. Collaboration noun, the act of working together with other people or organizations to achieve something. Spelling: COLLABORATION. The development of CRISPR-Cas9 as a gene editing tool was a result of a fruitful collaboration between Jennifer Doudna and Emmanuel Charpentier. Scientific progress often relies on international collaboration. Teamwork and collaboration are essential skills in many professions. Breakthrough noun, an important discovery or event that helps to improve a situation or provide an answer to a problem. Spelling: BREAKTHROUGH. The ability to program the CRISPR-Cas9 system was a major scientific breakthrough. Researchers are hoping for a breakthrough in the fight against Alzheimer's disease. This technological breakthrough will change how we live. Pioneer noun, a person who is one of the first people to do something important or to develop a new idea. Spelling: PIONER. Jennifer Doudna is considered a pioneer in the field of gene editing. Marie Curie was a pioneer in the study of radioactivity. Pioneers often face many challenges but open up new possibilities for others. Nobel laureate noun phrase, a person who has been awarded a Nobel Prize. Spelling: NOBELLAUREATE. Jennifer Doudna became a Nobel laureate in chemistry in 2020. The university invited several Nobel laureates to speak at the conference. Being a Nobel laureate is one of the highest recognitions in science and other fields. Practice these words. Try using them when you talk or write about Jennifer Doudna or related scientific topics. Let's further explore grammar by looking at more examples related specifically to Jennifer Doudna and her groundbreaking work. Talking about her journey and influences. Growing up in Hawaii, she was exposed to a rich natural environment which influenced her interest in biology. She has been working at UC Berkeley for many years. If she hadn't read The Double Helix, she might have chosen a different career path. Describing the CRISPR-Cas9 discovery and mechanism, the Cas9 protein, which is an enzyme, is guided by RNA to the target DNA site. Once the DNA is located, the Cas9 enzyme makes a precise cut. This system allows scientists not only to cut DNA, but also to insert new genetic information. Discussing the impact and ethical considerations, the discovery of CRISPR has raised important ethical questions. It is hoped that this technology will be used responsibly for the benefit of humanity. Governments and scientific bodies are discussing guidelines for the use of gene editing. These examples show how various grammatical structures are woven together to tell a compelling story of scientific endeavor, describe complex processes, and discuss broader implications. Jennifer Doudna's story is profoundly inspiring. It's a narrative of how fundamental curiosity about how life works, in her case, the intricacies of RNA and bacterial immune systems, can lead to discoveries that have the power to change the world. Her journey from a girl fascinated by nature in Hawaii to a Nobel Prize-winning scientist is a testament to dedication and intellectual passion. Her work also underscores the critical role of collaboration in modern science. The partnership with Emmanuelle Charpentier, bringing together their distinct expertise, was key to rapidly unlocking the potential of CRISPR-Cas9. It shows that even brilliant individuals can achieve more when they work together, share ideas, and combine their strengths. This is a valuable lesson for any field of human endeavor, including language learning, where interaction and mutual support can greatly enhance progress. Furthermore, Jennifer Doudna has been a prominent voice in the discussions about the ethical implications of gene editing technology. She has actively engaged in public debate and called for caution and responsible development of this powerful tool, emphasizing the need for societal consensus on how it should be used, especially concerning changes that could be passed down through generations. This demonstrates a deep sense of responsibility that accompanies great scientific power. Her achievement as a co-recipient of the Nobel Prize also serves as a powerful inspiration, particularly for women and girls in science. It highlights the extraordinary contributions women are making at the forefront of scientific research and discovery. Think about the qualities that define Jennifer Doudna's journey: intense curiosity, meticulous research, the courage to tackle big questions, the wisdom to collaborate, and a sense of responsibility. These are attributes that can fuel success in many areas of life. As you pursue your English language goals, let her story remind you of the power of persistent effort and the exciting discoveries that await when you are curious and dedicated. As we draw to a close, let's summarize the key takeaways from our exploration of Jennifer Doudna's life and work. First, we learned about Jennifer Doudna, the American biochemist whose passion for RNA and collaborative spirit led to her co-discovery of the CRISPR-Cas9 gene editing system. Second, we gained a simplified understanding of CRISPR-Cas9 as a programmable tool that can precisely edit DNA, revolutionizing biological research and offering immense therapeutic potential. Third, we reviewed important grammar points such as the past simple for recounting life events, the present perfect for ongoing impacts, the passive voice common in scientific discourse, and modal verbs for discussing potential. We also expanded our vocabulary with terms relevant to her field. And finally, we drew inspiration from her scientific journey, the importance of collaboration highlighted by her partnership with Emmanuelle Charpentier, her role in ethical discussions, and her achievement as a Nobel laureate. I encourage you to carry the spirit of curiosity and determination into your English studies. Just as Professor Doudna delved into the mysteries of the cell, you are delving into the intricacies of a new language. Every new word, every grammatical structure understood, every successful conversation is a step forward in your own journey of discovery. Embrace challenges as learning opportunities. Don't be afraid to make mistakes, and be confident in your ability to learn and communicate. Thank you for joining me on Grow English Mastery for this insightful session. What did you find most compelling about Jennifer Doudna's story or the CRISPR technology? Are there other inspiring figures or scientific advancements you'd be interested in learning about in English? Please share your thoughts, ideas, and questions in the comments section below. Your participation truly enriches our learning community. If you found this video valuable and enjoyable, please give it a thumbs up. It really helps our channel. And if you haven't already, please subscribe to Grow English Mastery and click the notification bell so you don't miss our future lessons designed to empower your English skills. Sharing this video with friends or anyone who might find it interesting would also be greatly appreciated. Keep learning, keep asking questions, and keep growing. The world of knowledge is vast, and English can be your key to unlocking so much of it. Until next time, take care and happy learning. Bye for now.